Good day everyone, welcome to the course on next generation sequencing technology, data analysis and applications. In the last few classes we have discussed about variant calling and we have seen hands on how to do variant calling using SAM tools and BCF tools, and we discussed the steps and then we also discussed how to visualize these mappings or alignment and how to verify some of the observations, some of the variants that we observe in the data. And then we started discussing the VCF file format where the output of this variant calling data is stored, right, and we have specifically discussed the data lines. So, in this class we will discuss the meta information lines and finish the VCF files, and then we will talk about something called variant annotation. Okay, so here is the agenda for this class. So, we will talk about VCF file meta information, and then we will talk about variant annotation, what kind of data we need for variant annotation, what we mean by variant annotation, and then the tools that we can use to do variant annotation. Okay, so let us start with the meta information lines. We discussed about the data lines and we saw certain fields in the data lines that we did not understand completely, right, and I said those information will be present in the meta information lines. Okay. So, in meta information lines, the lines will start with the sign double hash. Okay. We will see this in a moment when we look at the VCF file again carefully, and these lines will be written in key equals to value format. Okay. So, we will see again what do we mean by this key equals to value format. Means key will have some name and then value will be certain value. Okay. The first line usually is the file format equals to version number of VCF, which version we are working with, because they are updated and there are minor modifications in the format. And the next lines, this will contain the first thing will contain is source. So, the program that generated the files. Okay. So, here in this case, right, it should have the name of the program that actually we used. Then the next line will be reference, which is the reference genome. So, in this case it should be the is reference genome that we used. It will just take the file name and then it will have something like config or config assembly. So, this is actually identical to the SQ field from the SAM file. Okay. So, it should have these chromosome numbers or chromosome IDs in our data. Meta information lines should contain info, filter and format entries. So, we will see what these fields are. We have seen this info, filter and format in the data lines, okay, and these should also be present in the meta information lines. Okay. So, let us talk about info field first. In the info field, we will have the IDs that will be used in the info column of the data lines. Okay. So, we will have some IDs that will contain some information and the same IDs will be used in the data lines to convey some information about the variants. Okay. So, it will look something like this. So, it will start with this double hash info equals to ID equals to ID, number equals to number, type, description, source and version. So, this is how the info field will look like. Now, the first part is mandatory, right. So, this ID, the number, type and description, this should be there, and the source and version part is optional, they might not be there for the info fields. So, the ID is a unique ID. So, we will see some of these examples what this ID would look like, and then you have the number. This describes the number of values for each ID that will be present in the info field. Okay. So, for each variant, whether you will have one value or two values etcetera, this will be given here. Okay. So, again this helps us in understanding, right, how to parse this file. So, this usually this number is 1 2, right. So, 1 means we will expect one value for each variant, 2 means two values for each variant etcetera, but in some cases the value may not be known, right, the number of values that will be there. So, we have this dot sign. In addition, sometimes we can have special characters, okay. Instead of numbers or dot, we can have some special characters. So, these special situations are here, right. So, in some case we might have one value per alternate allele, okay. Then we will use the letter A for this number. So, you will see this number equals to capital A. Okay. So, then it means we will see one value per alternate allele. Now, this may vary, right, because if you have 3 alternate alleles, you would expect 3 numbers. If you have 2 alternate alleles, then you will see 2 numbers, right. So, that is how A is actually more relevant rather than specifying the exact number, because we do not know the number of alleles for each variant, right. So, this number of alleles can vary for each variant. You can have second situation where one value for each allele including the reference will be given, then we will use the letter R for number equals to R, and you can also have one value for each possible genotype, then we will also use the letter G. Okay. So, these are the possibilities that we have for the number field. What about then type? Okay. So, type actually describes the type of value that we can expect. Okay. So, this could be integer, right. So, in case of 1, 2, 3, right, this will be integer value, or it could be string, right. It could be float, right. If you are, let us say, talking about frequencies, right, fractions, right. So, that will be float, or it could be flag. Okay. So, we will see some examples and then it would be clear, right, what we mean by these different types. Okay. And then finally, you will have a description, right, that will tell us, okay, what this ID specifies, okay, what this ID means. So, it helps us in interpreting the data. So, here is an example. Okay. So, you can see this for example, the first line info ID equals to NS, right, and then you have number equals to 1. So, we expect one value for this ID NS, then you have type integer, this should be an integer value, and this description means number of samples with data. Okay. So, this is something how this format will be defined, that this info will be defined. Okay. Next, you have, let us say, for example, ID equals to DP, right, number equals to 1, type equals to integer, right, again description is raw, read, depth. Here is one example, you can see this number equals to A, capital A, right. So, this is one value per allele, right. So, that will be given, type would be float, and this description is allele frequency, right. So, as we just mentioned, if you are specifying frequencies, then you will probably use this type of float value, right. So, this is the type would be float. You also see this type flag, right, and then number is 0, right, because you do not expect number, but it is only a flag that will be used, and here the description is some membership of certain database, right, whether this variant is present in this database or not. If this flag is present, the variant is present in the database. If this flag is not present, the variant is not there. Okay. So, this is how the flag will be used. Okay. And similarly, you can see that a lot of these values, lot of these IDs can be there, and again you now understand how they will be specified. Okay. And these IDs will be there in the info field of the data lines. So, we have seen the data lines, right. So, you can now connect this information to the data lines, right, and see, okay, what these values actually mean for each variant. Okay. Then you have the filter field as you have mentioned, right. So, shows how the data is being filtered, right, and this is how it would look like, filter equals to ID and some description. Okay. So, what you have here are some examples from the files, right, from an actual VCF file. So, you can have filter ID equals to pass, description all filters pass. So, we have just mentioned this. So, if the variant passes all filters, then you will just use this pass in the filter field. But then we also describe this ID equals to Q10, right, quality score below 10, right. So, this actually is given in the description, you can see quality score below 10, right, then we use this Q10, right. So, if a variant does not pass this filter, we will just mention Q10, right. So, we had one example where the quality score was below 10, so we use the filter Q10. Similarly, we have this filter ID as 50. So, this description is 50%, less than 50% samples have data. Okay. Again, you can set these different filters, and for different types of analysis, etc., you can have different types of filters being set, and all those will be described in the meta information lines. Okay. So, as you can see, this VCF file can vary from one analysis to another depending on the types of filters, depending on the info that are given, right. So, that is why you need to carefully look into the meta information line to interpret the data lines. Okay. So, to understand more about the variants. Okay. So, then you have the format field as you have seen, right, and this format field in the meta information line, it describes the contents of the format column of the data lines, right. So, for each variant it will mention what are the IDs that are there for format and how the data will be arranged, right, how they will be arranged, this is also given. Again an example from a file, VCF file, you see this again, it starts with double hash, you have format, ID equal to GT, number equals to 1, type equals to string, description genotype. Again we are following the same process, right. We have some ID that is given, then you have the number, right, whether we can expect a number or it can be a G you can see, right. So, individual value for a genotype, right. So, you will have this, and then type is integer, description list of red scale, genotype likelihoods. Okay. So, again this will describe what you are what you are describing with these IDs. Okay. So, this description is very important to understand the data. Okay. So, then you have also have alternative allele field, right. So, this describes alternative alleles for structural variant, this is especially used. So, alt ID equals to type and description. Now, for SVs the type, so this ID will indicate the type of structural variant that you are looking at, and this is often a colon separated list, but the first type must be one of the following, like del for deletion, INS for insertion, dup for duplication, INV for inversion, CNV for copy number variation. You have also different subtypes and also can define more complex SVs, but we are not going into detail. There are specific ways to do that with the VCF file. So, you can see like VCF file is a very information rich file, and there is you have to understand, right, what actually you are representing or what is present in these files. Okay. So, you need to go through all of these information. So, coming back to the data lines, now we have understood the meta information lines. So, we can now look at these data lines and we can see, right, what this actually would look like, right. So, we have this chromosome position, right. So, again not chromosome number, some ID, position, then we have the ID, right, RS some number. So, this actually gives us an ID to some database, right, this variant is present in that database. Then you have the reference, alternate allele, quality, filter, right. Again now we better understand what this filter actually mean. Pass means all filters passed, Q10 means that this filter was not cleared by this variant, and you can see why. Then in the info we now know what this NS mean, DP means, A means or F means, right. So, all this information we have described in the meta information line, right. So, we can now connect this and we can understand what it means. For example, you can see this DB means this is this variant is present in certain database, so that this is a flag that is given, right. And then of course, you have lot such values after this, and of course, you have to then go back to the meta information and see and how to interpret this data. Then you have format, you can see certain parts here. So, GT, GQ, again these are defined in the format fields, right, and these again you can go back and see and connect with the meta information lines, and then you will understand what these data lines contain. So, hopefully, so this is clear, right, how we actually read VCF file by combining the meta information line and the data lines. This is where the file is open, right. These are the data lines that we have already looked at, and this is the header line. We have this chromosome, position, ID, ref, etc. And on top of that, right, if you go up now, I did not show you the meta information part, but on top of that you have the meta information part now. Okay. And the first line is file format equals to VCF v4.2, you can see that. Then you have the filter, right, ID equals to passed, so this will mean all filters passed. Then you have VCF tools version, so this is the program that generated, it also mentions the command that was used to generate this file, the reference file name that we used, right, here. Then as I mentioned, the contig names or the IDs of the chromosomes, right. So, these are also given, right, the lengths are given also. Then you have the alternate alleles, right. So, these are the alternate alleles that are present. Then you have the info part, right, you have this info part here, and here you have ID equals to indel, number equals to 0, type is flag, okay, whether it is an indel or not. So, there is not a, this is not a number, it is a flag, right. So, if it is an indel, there will be flag, indel flag, right, you will just mention indel, as you have seen, right, as you can probably see in some of the data lines visible here, right, you can see this flag indel, right. If this flag is present, means the variant is an indel, right. But if this flag is not present, then the variant is not an indel, it's SNP, right. As you can see here, this is an SNP. So, you have a lot of these now defined, right. We took some examples, but here you can see in our file there are a lot more info fields defined for you. Okay. So, you have, for example, IDV is the maximum number of raw reads supporting an indel, right. How many of the reads actually are showing this indel in the data, right. You have IMF, maximum fraction of raw reads that are supporting an indel, right. This is converting to fraction of the data. Then you have this DP, which is the raw read depth. Then you have lot of other measures as well. Okay. So, you can have some sort of test for base quality bias, for mapping quality bias, read position bias, etc. So, what these are actually looking at is whether the variant that has been called has been generated because of some biases in our data. Okay. There might be a lot of biases, so there could be some errors in calling. So, this is what is being tested by this test. Okay. So, that is what these different IDs are mentioning, right. And you will also mention which one is better, which one is actually good for you, right. And what you can do is you can use this information to set out filters, right. If you are interested, you can say, okay, we do not want these kind of biases in our data in the final variant call. So, that is why we will set out certain filters based on these biases that have been defined in this file. Okay. So, this is the info part, then you have the format part, okay, and there are only two format, right. So, you can see this. So, one is the list of rate scale genotype likelihoods, and then you have this genotype, only GT. Okay. So, here you will not have this haplotype quality because we are dealing with haploid genome, right. So, it is not a diploid genome. So, you do not have to do phasing, etc. All right. So, then you also have some other ones, right. You can see some other info fields. You have this ID equals to AC, ID equals to AN, right. Again, there would be descriptions saying what this is doing, right. And again, according to that, you have, for example, number A. So, you have a value for each alternate allele, right. So, this is something that is mentioned again here. Okay. And then for the other ones, you can see the numbers. So, 1, 4. So, for every variant, you will have 4 values or 1 value, etc. So, this is defined here. And then finally, it ends with also the BCF tool call version again, and the final call command that was used for BCF tools. Okay. Now, let us look at this right side now. Okay. So, this part, the first part of the data lines, those we have discussed, right. So, here the ID we do not have any mapping to any specific database. So, the ID field is empty, right. We have not set out filters, right, except this Q20. So, that the filter field is also empty, and the info here, right, that is populated with all these numbers here, you can see, and then you have the format, and then you have only one file here. So, this file names, the sample name is given final file.bam. So, if you have multiple ones, those will be listed one after another. Okay. So, let us see, right, you can see this, right, for this. So, the first you have these info fields, and for each of them you have these values given. Okay. You can see them here. So, these values are given, and again this follows the meta information line. So, meta information line says, okay, this SGB, what this stands for, and it should have one value, right. So, that should be float or something, and that will be followed here. Okay. And similarly, after that you have this format part. So, this format part specifies how this will be defined for the samples, right. So, this is the order, and again this has been defined in the meta information line, and the information is for this final file.bam. So, imagine if we had multiple ones, let us say final file 1.bam, final file 2.bam, etc. Those will be repeated one after another. Those will be present in the same format one after another. Okay. So, now I hope it is clear, right, how you can combine this meta information line and then with the data line to understand the full BCF file and what is actually represented in this file. Okay. So, this is done. So, the next step is to actually go for variant annotation. Okay. So, we will go back to the presentation and we will talk about variant annotation. Okay. Okay, so we take the VCF files and we have seen this meta information line, the data lines, and we can set up certain filtering schemes, right. If we say we do not want certain biases in our data in our final calls, then we can set up certain filterings based on these fields that we have just described. Okay. There are many fields and maybe some of them could be useful for certain analysis. And once we have done this filtering, we can do something called variant annotation. Okay. So, what is this variant annotation? We will discuss this in detail now and why is this useful. Okay. So, for any variant call, you have to interpret, right, what this variant actually means and whether there is any significance of this variant in your data. Okay. So, what we are doing by annotation is we are measuring likely impact of variant on function, maybe gene function or cellular function and cellular phenotype, right. So, we can probably map it to certain genes, etc., or we can, or we are doing known association with phenotypes, disorders or diseases. Okay. So, whether this variant has been seen in other individuals and has been associated with certain diseases, for example, genetic diseases or so, right. So, this kind of annotation we can do. So, there could be many possibilities. So, a single nucleotide polymorphism variant, this could be in the promoter region of a gene, right, and it can thus regulate gene expression, right. So, you can say it can affect binding of transcription factors and it can impact the expression of the downstream gene, right, or a SNP variant can be synonymous and or non-synonymous or nonsense. So, synonymous means there is a changes in the coding DNA sequence, but no change in the amino acid sequence. It could be non-synonymous, means there is changes in the coding DNA sequence leading to changes in the amino acid sequence. Okay. So, these variants are likely to have functional impact. Okay. And on top of that, you can have nonsense mutation. Okay. So, this nonsense mutation refer to introduction to of premature stop codon in the gene, and this can lead to loss of function. Okay. So, you can think of many different other type of annotation, right. We have just discussed two types of annotation. You can have regulatory function. So, a variant that you have found, it might be regulate, it might actually change expression pattern of a gene. So, it could be very important there, or it could be changing the function of a gene, right. You can, it can lead to loss of function or maybe change the function in certain ways. But you can also have other types of variants, right. For example, it can affect, let us say, folding of a protein, right. You can think of splicing events, right. You can have variants that change splicing events for a gene, right. All those things can be there. So, these will be done through this kind of annotation. Okay. Now, how do you do this annotation? Okay. So, for doing this annotation, we need something called GFF or GTF files. Okay. So, GFF and GTF files, so GFF stands for general feature format and GTF stands for general transfer format, which is actually same as GFF version 2. So, we will discuss only the GFF format and that should be enough for doing this variant annotation. Okay. So, let us talk about the GFF file, what it contains, what kind of information you have. It is actually quite simple compared to other ones that we have discussed, the VCF or the SAM, which are much more elaborate. So, GFF is much easier to interpret. So, here is some example you can see. So, here you have on top, you again have some headers, again you have this hash or double hash, right, and it tells us that which GFF file we are looking at, which is the species, right, the genome build, what is the GFF version, etc., right, and where did we get this data from, what is the assembly version of this genome, etc. So, here we have this information, okay, and then you can see these numbers, these IDs on the left, right. So, these NC numbers, right, these are the IDs of the chromosomes, then you have the source, then you have some other information. Okay. So, we will discuss what these fields are actually just in a moment. Okay. So, each line is a feature in the genome and this actually is represented by 9 fields. Okay. So, these 9 fields are tab separated and the empty fields are denoted by dot, as we have seen for other files as well. So, if you do not have information, you just put this dot. Okay. So, what are these file fields, right? So, if you want to annotate your variants using this GFF file, you need to know this because you can probably write a code that will do this for you, right. Some of the very simple annotations can be done by yourself. Okay. So, what are these file fields that you have? So, the first one is called sequence name, is usually the name of the chromosome or the scaffold or a valid identifier. So, in our case the example that I have shown you, it is an identifier from a database, right. So, this is again pointing to a chromosome, right. So, instead of that identifier you can directly name the chromosome there. Then the second field is a source, right. So, this is the name of the program that generated that feature or the source database, right. So, here in this case you can see in the example it is the refsec database, right, from which we obtained the GFF file. The third one is very important, is the feature itself. Okay. So, what is this feature actually we are talking about? Is it a gene? Is it an exon? Is it a variation? Right. What type of feature we are looking at? Okay. So, we will look at the example again and you will probably understand this better. The fourth is the start position. Okay. Where does this feature start in the genome, and this follows the 1 based positioning system. Okay. So, we talked about the 1 based and 0 based positioning system. GFF follows the 1 base, right. So, starting base is 1, numbered 1. Then you have the fifth field, which is the end position, where the feature ends, right. So, if it is a gene, what is the location, right, whether it is from 100 to 150, 200 to 700, etc., right. So, this is what this start and end position will give you, right. Again this follows the 1 base position system. The next one is score, right. So, this is a value that indicates how confident the source is about the annotation. Okay. So, whether it is a gene or an exon, this confidence will be reflected in the score in the sixth field. So, if the value is not available, there is no confidence value, it is just represented by dot. Okay. Then you have the next one, right. So, it is the strand field, which is defined as the plus or minus. So, you probably can understand whether the feature is present in the plus strand or the negative strand. The eighth one is the frame. Okay. So, this is actually important if your feature is present in the coding sequence, right. So, it could be 0, 1 or 2. So, 0 means that first base of the feature is the first base of a codon. Okay. So, I think you can probably now relate whether this is likely a synonymous mutation or a non-synonymous one, etc. Those you can find out if you know this value, right. So, 0 means the first base of the feature is the first base of a codon, 1 means the second base of the feature is the first base of a codon, and so on, right. And the final field is the attribute. Okay. So, this is a semicolon separated list of tag value pairs, and it contains additional information about each feature, right. So, it will have some description kind of thing. Okay. So, let us go back to this example again, right. So, here is your identifier, this is the chromosome ID, then you have the source, then you have the feature name, whether it is a gene, mRNA here, exon or coding sequence, CDS, right. This is given here, or in here you can also see the telomere, right. So, where is the telomere region? So, the positions are given. Then you have the start and the end positions, right. So, for this gene, of course, the gene, mRNA and exon, they are following the same start and end, because there is no multi-exon gene. So, this is not a multi-exon gene, right. So, then you have the dot. So, this is okay. So, start and end, then you have the score, right. This is the confidence, right, if we remember correctly. Then you have the strand, okay. This is the strand plus minus, and then you have this the feature, whether it is 0, 1, 2, right, whether it starts with the codon, right, whether it is starting with the codon, etc., right, that information is given. So, it is again not given for many of these features, but for some, which is the coding sequence CDS, this feature is given, right, 0, right. This is the starting position of the codon, right. Then you have some additional attributes, right, as you can see again some extra information. So, here is the gene name. So, this gene name is given, or in case of CDS, this is the CDS ID, again this is the identifier that is given, and you can also see a lot more information. Okay. So, if you go to the original GFF file, you will find a lot more information about these things. Okay. So, just to again highlight, this is the sequence name, this is the source, this is the feature, start, end, score. this is the strand and this is the frame, right, 0, 1, 2, and then you have the attributes here. Okay. Now, the second type of annotation that you can do is associate these variants with phenotypes, disorders or diseases, right. So, how we do that? This is very simple actually. So, people have collected or researchers have collected this data over many years from many patients, etc., from many studies, and they have put them into the databases. Okay. And what we need to do simply is to analyze whether the variant that we have found is present in these databases of variants. Okay. And this is very important, especially for human data. If you are working with human variants, right, human samples, you want to check this. Okay. And there are several databases. I will just mention some of them. Here is one. This is a very common one. So, it is called DBSNP, right. So, it contains a lot of these human signal nuclear variants, microsatellite information, etc., and small indels. Okay. So, you can check if you have this variant data in your case, you can check whether this is already reported in DBSNP, is a common variant in humans or not, and whether there are some clinical association. So, this information will also be present. Okay. Then you have something called the OMIM database, it is the online Mendelian inheritance in men. So, here again this is a online catalog of human genes and genetic disorders. So, again looking at some of the variants that have been associated with genetic disorders or diseases. Okay. You can so you have also more recent databases coming up, right. So, for example, this is one database where you have a lot of this comprehensive variation annotation. So, it is not just this clinical annotation, etc., it also contains information about regulatory information, whether this variant is associated with certain gene regulation changes, whether there is some sort of change in transcription factor binding. All those informations now can also be combined together and put into the database. So, just a few words about the variant annotation tools. Okay. So, here are again some common ones. Of course, there are many more out there. The first one is called Annovar. So, again you can go and check in the link. So, you can have gene based annotation or region based annotation, looking at whether it affects certain, your variant affects certain function of certain genes, or they are present in certain regions which might change the gene regulation, etc. You can also have another tool called VAR-AFT. So, this is variant annotation and filtration tool. So, this actually helps in annotation using DBSNP or OMIM if you are working with human data. It also associates your variants with gene ontology and certain biological pathways. We will talk about gene ontology in later classes in much more detail and see what this actually contains. So, it mostly looking at the functions, etc., or involvement of genes to biological processes. Okay. So, these kind of tools are now available and that you can utilize for your variant annotation. Okay. So, here are the references that we have utilized for this class. So, just to summarize, we have talked about BCF files, the meta information lines, and it kind of gives us a complete picture of the data lines, right, what kind of information we are getting in the data lines. Then we talked about the variant annotation part, right. So, we are looking at possible impact of variants on gene function and phenotypes, and as well as we are trying to associate them with disorders and diseases if they are known already. And we talked about some of the variant annotation databases that can be used and will be useful if you are especially working with human data, and we also talked about some variant annotation tools that can be utilized. Thank you.